Hi everyone. This is the adaptive immunity lecture. In this lecture, we're going to learn about the second part of the immune system. So we've already covered innate immunity, and now we're going to learn about adaptive immunity. In this lecture, we're going to cover the basic processes of how you actually activate adaptive immunity. So adaptive immunity is a little bit more involved than innate immunity. And within adaptive immunity, there are two components. There's humoral immunity and cellular immunity. So you're going to learn about both. And then finally, we're going to end the whole lecture by talking about the immune system in bacteria called CRISPR. So just to give an overview again, your immune system, as we talked about in the last lecture, consists of two components. So everyone, their immune system, they have innate immunity and adaptive immunity. And we've already covered innate immunity. Within innate immunity, we said that your first line of defense are things like your skin, secretions that we make, saliva, oil, sweat, mucous membranes, and the microbiota that protects you. So the innate immunity, again, is always on. Your skin's always there protecting you. And then for some reason, if a pathogen evades these things, then your internal innate immunity defenses kick in. So those are different white blood cells that are phagocytic, natural killer cells, defense proteins. So we talked about the complement system. We talked about antimicrobial peptides, and we talked about inflammation and fever. So that was innate immunity. This is the one that you're born with. Remember, that's why it's called innate. You innately have that immune system. It's always on. It's when we say that innate immunity is non-specific, we mean that it helps protect you against any pathogen. Now, if this immunity somehow fails you, your adaptive immunity kicks in, and it's called adaptive immunity because it adapts over time and it gets better. And it's activated by continuous exposure to different pathogens, so it gets better with time. These are where vaccines help, and we're going to talk about that a little bit later. So again, your immune system consists of innate and adaptive immunity, and today we're going to focus on your adaptive immunity. So the adaptive immunity is always continuously developing throughout your life. Let me put all of these out. And it's the third line of defense. When we say third line of defense, we're assuming that your innate immunity, your first line of defense is like your skin and the external factors. Second line of defense are like inflammation, fever from your innate immune system. And then the adaptive immunity is the third line of defense. And its response is a little bit slower than innate immunity, so it takes several days to kick in after innate immunity hasn't responded. And here we're talking about the white blood cells that are involved in defending you against pathogens and their lymphocytes. It's strictly internal, unlike innate immunity where external factors are like skin and secretions, and it has memory. So adaptive immunity, again, it continuously develops over your life. And when I say it has memory, I mean that, for example, like if you get exposed to one strain of influenza, the idea is that your body is going to build antibodies against that specific strain, and so hopefully you'll never get sick with that strain again. And this is the same idea that people are researching now with coronavirus patients. So the question is, do people who've been infected with SARS-CoV-2, will they get sick again? There's been a lot of studies studying this. I guess people are not really sure yet, but we do know that when we look at patients that have recovered, we are seeing antibodies, and you can take those antibodies and hopefully give them to a patient who hasn't built that immunity yet. So this is an overview of adaptive immunity. Adaptive immunity, the specific white blood cells that it deals with are lymphocytes. And this lymphocytes, remember, there's B lymphocytes and T lymphocytes. Now, I want to tell everyone that B cells and B lymphocytes are the same thing. T cells and T lymphocytes are the same thing. And I'm going to use both terms throughout the lecture so that you become comfortable with me saying T cells or T lymphocytes. They're the same thing. And these are the specific white blood cells that recognize pathogens and promote adaptive immunity. So it takes weeks or more to build adaptive immunity following the first exposure. Usually you get very sick initially. That's your primary exposure to a pathogen. And then If your adaptive immunity with time will remember that you got exposed to that pathogen, it'll adapt to it. And then if you get exposed again to that same pathogen or microbe that caused disease, the secondary response of your immune system is going to be stronger. So you're going to fight things off in a stronger way. So an example of this, I'm going to use vaccines as an example. Here an example is vaccines usually are subunits of microbes that will not make you sick. So for example, if you think of polio, we take the polio vaccine. So you get a component of the virus and you get the shot in the form of a vaccine, and your body builds antibodies. So that's your primary response. In a way, you're not getting polio, but it's an artificial way of building adaptive immunity. So then if you're ever in your life exposed to polio and you actually get the viral infection, your body already has a secondary response. The antibody is there to deal with it. So this is why adaptive immunity gets better with time. And again, the cells involved in adaptive immunity are lymphocytes, so B lymphocytes and T lymphocytes. And you're going to see this image throughout my lecture. We're going to focus on both B cells and T cells. The idea is that B cells attack outside invaders, so extracellular bacteria. T cells attack intracellular pathogens like viruses. So viruses, if you think of a viral infection, that's where T cells will be involved. If you think of a bacterial pathogen that's floating in your blood, that's where we will see B cells to defend you. And they're both lymphocytes. And adaptive immunity, just to add another layer of complexity, has two components. So when we look at, so to start off, you have your immune system. Your immune system consists of innate immunity and adaptive immunity. And then when you look at adaptive immunity, it can further be split into two different immunities. And adaptive immunity consists of your humoral immunity and your cellular immunity. And together they make adaptive immunity. Humoral immunity eliminates extracellular pathogens. So this is anything that's like floating in your blood, different toxins, anything that's not getting in your cells. This is called an extracellular pathogen. And these are bacteria, toxins, viruses in the bloodstream, tissue fluids, anything like that. So in the bloodstream fluid, tissue fluid. And humoral immunity involves B cells or B lymphocytes. That's why I'm saying B cells or humoral immunity has to do with helping defend you against pathogens that are extracellular. And with B cells, the reason why this is called humoral immunity, humoral comes from fluid, so that's where the term comes from. It means things that are in fluids, like your bloodstream. So again, humoral immunity eliminates extracellular pathogens, but if you get infected with an intracellular pathogen, like a virus that infects one of your cells, maybe like a specific hepatitis virus that infects your liver cells, this is where your T cells kick in, and we call that immunity cell-mediated or cellular immunity. So this is the one that kicks in when we have pathogens that are intracellular inside a cell. So an example of that are virally infected cells. This is where we see cell immunity. We also can see this if we have, there are some bacteria that are intracellular bacteria that cause infections. So anything intracellular, this is where your cell-mediated immunity kicks in. And this is the immunity that also attacks cancer cells. People with cancer, or even people with not, we all have some cells in us that are mutating all the time. So with cancer cells, it's not like your body is not doing anything. Your body is fighting it off. Your immune system is fighting it off, but you still need to take, if you do have it, you still need to take medication, but your body, your immune system is still playing a big role in helping fight off cancer cells. And cell-mediated immunity involves T cells or T lymphocytes. So again, to summarize, because this is going to keep coming, adaptive immunity has to do with humoral immunity and cellular immunity. Humoral immunity is the one that involves B cells or B lymphocytes, and this is the immunity that protects you against extracellular pathogens or pathogens that are outside cells, like the bloodstream, toxins, viruses, anything that's outside cells. And adaptive immunity also has cellular immunity, which has T cells that protect you against intracellular pathogens or pathogens that infect inside cells or cancer cells. So keep that in mind as we go through the lecture, because we're going to give a lot of detail on humoral immunity and cellular immunity. So again, adaptive immunity has to do with lymphocytes. Humoral immunity involves B cells or B lymphocytes. Cellular immunity involves T cells or T lymphocytes. And humoral immunity is where B cells attack outside invaders, and cellular immunity is the one where T cells attack intracellular pathogens. And both B cells and T cells have memory cells. That's why we say adaptive immunity gets better over time. So your immune system, you build it over time. This is why we previously said that like a 1-year-old doesn't have a great immunity because they haven't seen a lot of pathogens in their life compared to like a 20-year-old who's been exposed to a lot. They have a better adaptive immune system. And just to give a little bit brief knowledge about lymphocytes, when we talked about the blood smear lab, I did tell you guys that hematopoiesis is blood cell formation, and that happens in the bone marrow. So in the bone marrow is where all blood is formed. All leukocytes, all white blood cells are formed. Remember, there's many different types of white blood cells or leukocytes, but one type of white blood cell are lymphocytes. So I want everyone to know that they're formed in the bone marrow and stem cells. Again, bone marrow is like if you take a bone, you cut it and have that inside material, that's called bone marrow. Also, all blood cells develop there, including lymphocytes. B cells are called B cells because they develop there and they mature in the bone marrow, the same area. But T cells develop in the bone marrow where all blood cells develop, but they mature in the thymus. That's where the T comes from. And once mature lymphocytes go to lymphoid organs, which are the different organs that have a lot of immune protection. I'll talk about them in a second. And they wait to encounter a pathogen to help protect you. So in lymphoid organs, we're going to see a lot of B cells and T cells. And remember, B cells are the ones that are helping you against extracellular pathogens. T cells are helping you against intracellular pathogens. And again, we said B cells and T cells are in your lymphatic system. So your lymphatic system, I know most of you have taken anatomy, but it consists of tissues and organs. And when we look at the lymphatic system, it has a lot of B cells and T cells roaming around. So here, there's this fluid called lymph. And the benefit of lymph is you have immune system cells, white blood cells that are there to protect you. And each lymphocyte, the B cell or T cell, will be floating around. There's these little areas called lymph nodes that we have throughout our body. And in this picture, you can see where lymph nodes are. Like here, you have your tonsils and you also have lymph nodes. This is where we see a lot of T cells and B cells waiting to encounter a pathogen so that they can take it, attack it, destroy it in one way or the other to protect you. And there's also secondary lymphoid organs. These are things such as your spleen, your tonsils, your appendix, and there we see a lot of lymphocytes that are there to protect you. There has been some research on, do people who have their tonsils out have a worse immune system than people who still have their tonsils? So there's a lot of studies on that. And I think that it's potentially yes, people who have their tonsils potentially have a better immune system than those who don't, but it's very minimal because you still have a lot of other lymphoid organs to protect you. And again, lymphatic vessels, so these are these right here, you see them. These are not blood vessels, they're different things. They carry lymph fluid collected from the tissue throughout the body, and then the lymph fluid has potentially antigens or pathogens. We're going to talk about what antigens are in a second. And there we see a lot of B cells and T cells or lymphocytes in general, and they're there waiting to pick up pathogens and protect you against them. B cells and T cells go to lymph nodes. They're in different lymph organs. And again, their purpose is to capture any pathogens or antigens and bring them in contact with your humoral or cellular immunity. So we're going to define what antigens are right now. Antigens, the definition of them is they're antibody generators. That's where the word antigens are. And they're on they can be a lot of things, but they're molecules on pathogens that identify them as foreign molecules. So if we look at a bacteria or a virus, they look different than your cells. A virus is not a cell, we know that, it's not alive. But if you look at the a virus infectious agent and a bacterial cell, they have different things on them that make them look different than your own cells. So we call those antigens, and they're recognized by your immune system, and your immune system generates antibodies to target antigens. So antibodies are good things, and antigens are antibody generators. They're usually proteins or different sugars, polysaccharides, on the surfaces of pathogens or foreign cells. And they tell your body, like for example, if a bacteria goes in, the peptidoglycan from the bacteria is foreign. Your body knows that this does not look like its own cells. So it's going to generate antibodies to target the sugar antigen. And this is also the reason why when you give someone blood, we're going to talk about blood later in the semester, if someone is type A blood, you don't give them type B blood, because if you give them type B blood, their body is going to look at B cells as foreign, and they're going to create antibodies, and then their body's attacking the blood that you gave them. So that's what antigens are. And antigens can be components on bacteria, viruses, molecules from mold spores, pollen, house dust, different things that people are allergic to. So if someone is allergic to peanuts, peanuts have different parts that their body is seeing as an antigen, and their body is creating antibodies to fight that off. And their antigens are generally very large molecules. I don't want to stress too much about this, but when you have an antigen like a cell wall, in the antigen, there's different parts that the antibody will target. So we call those antigenic determinants or epitopes. So epitopes are like pieces of antigens. So again, if we think of a bacterial cell, and here is the antigen, let's say the cell wall there is the antigen, your body will create antibodies that will target different parts of the antigen that we call epitopes or antigenic determinants. The take-home message of this are pathogens create antigens in your body, and your body creates antibodies to target antigens. And this is a good video summarizing lymphocytes and antigens and how they go on to create antibodies.
Antigens are macromolecules, usually of molecular weight greater than 10,000, such as proteins and polysaccharides. They are recognized by the immune system as foreign. Individual antibodies are not made against the entire antigen molecule, but rather to particular chemical groups on the molecules known as antigenic determinants or epitopes. Many different antibodies can be made against a single antigen, each antibody reacting with a different epitope. Complex structures, such as the surfaces of bacterial cells, may have many different epitopes. Each different antibody binds only to the correct epitope.
So again, to summarize it, that your body creates antibodies that are targeting the pathogen by binding to its antigen. And to give a relevant example, so we talked about the cell structure of the SARS-CoV-2 virus, the one that causes COVID-19. When we look at the virus structure for coronavirus, it has spikes. Those spikes, when your body, if you get infected with it, your body recognizes those spikes as a foreign molecule, and so those are the antigen for the virus. The spikes are the antigen, and so your body's creating antibodies to target that antigen so that you don't get the viral infection, or that your body can clear it. And just a few more terms before we get into humoral and cellular immunity. When we look at B cells and T cells, they have many receptors. So if you look at the cell surface, they have many different protein receptors, and their function is to bind to different antigens. Receptors are very specific to the antigens they bind. So every cell has hundreds of thousands of receptors on them, and each receptor is there to detect one type of antigen. So they're very specific. So if you have a receptor that's targeting some antigen in the influenza strain, that's all it will target. It will not protect you against like infection by coronavirus, because they're different things, and the receptors are very specific to the pathogen's antigen. B cell receptors form specific antibodies. So when we look at a B cells have antibodies on their surface. We'll talk about them. They're the ones that create antibodies. T cell receptors, remember, they're there to recognize intracellular invaders. So they cannot see extracellular invaders or antigens. They can only recognize intracellular pathogens. So here's a B cell, here's a T cell. And for T cells, they're receptors to recognize an antigen. It has to be a pathogen that infected intracellularly, and then its components are displayed. And we'll talk about how that's done. So again, to summarize, because now we're going to get into the details of everything, adaptive immunity has to do with cellular immunity, here's cellular immunity, and humoral immunity. Humoral immunity involves B cells, and B cells are there to protect you against extracellular pathogens. Cellular immunity has to do with T cells, and T cells or T lymphocytes are there to protect you against intracellular pathogens or invaders. As an overview of B cells or humoral immunity has to do with B cells. B cells travel to lymphoid organs and they wait there for potential invaders or pathogens, or we can say antigens. They target extracellular pathogens or the antigens on the pathogens. These are examples of circulating bacteria or toxins or foreign red blood cells. So if you have a bacteria right now in your bloodstream, which would be a terrible thing if you have a bacterial infection or a toxin infection, B cells are there to target that pathogen or that extracellular antigen on the pathogen. Same thing with foreign red blood cells. So as a summary, B cells develop and mature in the bone marrow, and that's where they gain specific B cell receptors. They recognize antigens, which are pathogens or foreign molecules, and then they proliferate and differentiate into plasma cells. Plasma cells are B cells that make antibodies. So we call them plasma cells. So plasma cells produce antibodies. You can say B cells produce antibodies, but to be correct, it's actually effector B cells or plasma cells. That's the type of cells that should say plasma, not plasms. And antibodies, they're a great thing that our immune system creates, our humoral immunity, because they bind to the pathogen's antigen. And this way, when we're going to learn about when the antibody binds to the pathogen, and we're going to talk about antibodies later, but I do this because they look like a Y. They target it for destruction. And then you also have memory B cells formed so that if you ever encounter that extracellular pathogen, again, your body is ready to respond. So this is humoral immunity. With cellular immunity or cell-mediated immunity, this one has to do with T cells. T cells are in lymphoid organs, lymph nodes, tonsils, spleen, appendix, and they're waiting for intracellular invaders or cells that have already been harmed. Because remember, they cannot recognize antigens or pathogens that are floating outside cells. They have to be inside cells. That's what cell-mediated immunity is. They target intracellular antigens, so virally infected cells, cancer cells, transplanted tissue. We're going to learn about two sets of T cells that help protect you, and they differ in surface proteins that are called CD markers. So that's why if you're reading the textbook, you'll read about CD4 and CD8 cells, which are cytotoxic T cells and helper T cells. Cytotoxic T cells, from the name, they are toxic, so they destroy or kill host cells that have been infected by viruses, which is in a way a good thing. Helper T cells are another type of T cells that form, and helper T cells are probably the most important cells in our immune system because they go on to activate everything in adaptive immunity. We're going to learn about it. So the two subsets of T cells are helper T cells and cytotoxic T cells, and they have to be activated before they can multiply. Dendritic cells are white blood cells that do phagocytosis, and what dendritic cells do is when they see a pathogen, they eat the pathogen, and they present it on their surface. And in this way, we get T cell activation. We're going to learn about it in a second. Once T cells are activated, they proliferate and differentiate into effector T cells and cytotoxic T cells. So there's two groups of T cells. Again, there's helper T cells and cytotoxic T cells. Helper T cells go on to activate a lot of things in the immune system. They also go on to make macrophages stronger, which are good if you have an infection, because remember, macrophages are important in phagocytosis. And then T cells can also differentiate into cytotoxic T cells, which kill infected cells or cause apoptosis, cell death. So we'll learn about that in the end. A little bit more about cell-mediated immunity. Remember, all blood cells develop in the bone marrow. T cells are called T cells because even though they develop in the bone marrow, they mature in the thymus, which is a lymphatic organ. T cells recognize antigenic peptides processed from phagocytic cells. So for example, a dendritic cell is a phagocytic cell. They'll recognize the antigens that the dendritic cell is presenting, and T cell receptors on the T cell surface contact the antigen. So they contact the antigen that's been presented by the phagocytic cell. And this causes the T cells to secrete cytokines. Remember, cytokines are signaling molecules. We learned about them in innate immunity, which activate CD8 cells, which are cytotoxic T cells, and activate macrophages. Macrophages are good because they do phagocytosis, and you also get memory cells formed. So cytotoxic T cells respond to intracellular antigens, viruses, different types of cancers, and helper T cells activate B cells, macrophages, and they activate a lot of other things. So this is where in both humoral immunity and cellular immunity, I ended by saying that you form memory cells. So this forms the whole adaptive immunity. It's adapted to what it's been exposed to, and it gets better over time. Now, for both components of adaptive immunity, whether that's humoral immunity or cellular immunity, to kill the pathogen or target the pathogen, they need to be activated, and they're activated by helper T cells. So we're going to go on this level. Adaptive immunity is activated by helper T cells. Helper T cells, the reason why they're called helper T cells, it's because they help activate other adaptive immune system cells. They themselves need to be activated to activate B cells and T cells. If you think about it this way, we have T lymphocytes or T cells in us right now. There's no need for these cells to be active all the time, to use up your energy. It makes sense to activate or turn on your immune system when you actually see a pathogen or an antigen of a pathogen. So helper T cells are activated when they actually encounter an antigen presenting cell, or an APC. An antigen presenting cell is a cell that presents antigens on its surface. So this is what happens in the course of an infection. This is really simplifying it. You get a pathogen in you. Let's say, actually this is a not really a good example, but it makes sense right now. Let's say someone has SARS-CoV-2 and it causes COVID-19 and they're having active symptoms. And remember, transmission is through droplets. Let's say they sneeze in your face and then you breathe that in. Now the virus is inside you. The virus will be taken up by one of your white blood cells, which is a good thing. And that white blood cell will chop up different parts of the virus and put it outside its cell. Now that cell, your cell, becomes an antigen presenting cell, and it floats around. An example of that is like a dendritic cell. It encounters the pathogen, and now it takes it, it chops it up, and it puts the marker of the antigen, the pathogen that infected you, outside the cell, and now it's become an antigen presenting cell. So it's basically telling your body, hey, there is a pathogen infecting, I'm telling you guys something is not right. And now that antigen presenting cell is what will activate helper T cells. And helper T cells will go on to either activate humoral immunity, B cells, or cellular immunity, T cells. So this is how the whole activation works. And when the antigen presenting cell interacts with the helper T cell becomes activated, and helper T cell will go on to activate or help B cells or T cells. And then the B cells or T cells will target the pathogen for destruction. So the way to summarize is you have a pathogen in you. A pathogen has antigens on it or inside of it, whatever it is. Your cells will process the antigens and they will present it, your white blood cells. So then that becomes an antigen processing cell. That will cause a helper T cell has receptors that will interact with the antigen presenting cell, and this will activate the helper T cells. And now the helper T cells will go on to either activate humoral immunity if you've been infected with an extracellular pathogen, or cellular immunity if you've been infected with coronavirus is an intracellular pathogen. If you've been infected with that, you have your cell mediated immunity on so that you can turn on cytotoxic T cells that will attack virally infected cells. This is how it works. And here's a good video.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces.
So in this example, let's say this was like a whole human, not just a cell, and they ingested a food, let's say food that was contaminated with bacteria. So there's the food, and now the bacteria is in you, and your body recognizes, hey, this is a foreign molecule. So a macrophage, your white blood cell, because macrophages are white blood cells, will go and take in that bacteria. And there's more detail to this, but to simplify, they'll chop it up and they'll present little pieces of the bacteria. So now you have little macrophages or dendritic cells, because both are phagocytic cells, that have phagocytosed or ate the pathogen, and they chopped it up and they're presenting antigens on their surface telling your body, we have a foreign invader, we need to actually do something about it. We need to turn on the immune system to start targeting it.
Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1.
Okay, so your antigen presenting cell, so in this video they have the macrophage as an antigen presenting cell, but it could be a dendritic cell too, now interacted with a helper T cell, because we want to try to activate those helper T cells. So you have a helper T cell, the antigen presenting cells, they interact. Now what happens is the antigen presenting cell is going to release a chemical that will activate or turn on the helper T cell. That is a cytokine. I'm not asking you guys to memorize the specific types of cytokines, but cytokines are signaling molecules. We talked about it in the last one. Interleukin-1 is a type of cytokine. So let's just, to make it easy, we're just going to say it's a cytokine. It's a signaling molecule that goes on to activate this helper T cell so that the helper T cell can go on to either turn on humoral immunity or cellular immunity.
Stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells.
Cytotoxic T cells are involved in cellular cell-mediated immunity. B cells are involved in humoral immunity. B cells target extracellular pathogens. Cytotoxic T cells target intracellular pathogens.
Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells.
Which make antibodies.
These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may
Make a lot of
survive for several
antibodies.
decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
So again, to summarize, nothing is turned on until helper T cells are themselves turned on or activated. They're turned on or activated by an antigen presenting cell. An antigen presenting cell is a cell that took up a pathogen or the antigen and presented those pieces of the pathogen on its surface. And then their activated helper T cells. Helper T cells will go on to either activate B cells in humoral immunity or cytotoxic T cells in cellular immunity. And we're going to talk about
Active
the details of these. So this is what I just said. And we're going to focus now on talking about humoral immunity, and then we'll talk about cellular immunity. So with humoral immunity, B cells have to first find an antigen which matches its receptors. So remember, B cells have a lot of receptors on their surfaces, and they're very specific. B cell will wait until it's activated by a helper T cell. If it gets activated by a helper T cell that was activated from before, the B cells will divide to form B cells that will make antibodies, which we call plasma cells or effector B cells, and memory B cells. And the plasma cells are very important, those specific effector B cells, because they produce antibodies that will attach to the pathogen and destroy it. We're going to talk about what antibodies do in a second, why they're a great thing. And so pathogens marked with antibodies can be destroyed in many different ways, not just by phagocytes. And if the same pathogen infects again, memory B cells are there to help activate the immune system much faster. This is the same idea with vaccination. So the first step in humoral immunity, or in this whole immune system process is antigen recognition, processing, and presentation. So an antigen binds an antigen presenting cell, and an antigen presenting cell could be like a macrophage or a dendritic cell. It's a phagocytic cell. The antigen presenting cell that has taken up the pathogen will degrade the antigen into small fragments and will put it on its surface on a receptor called MHC2. Then the antigen presenting cell, let me go back, the antigen presenting cell with the antigen fragment on its surface will migrate in your body presenting the antigen so that your body can say, okay, we need to turn on the immune system. And it's presenting the antigen on MHC class 2 proteins that we have. Then the next step is helper T cell activation. So a specific helper T cell will bind this antigen antigen presenting cell that has the antigen on the MHC2, and the helper T cell will get activated because the antigen presenting cell so you have the antigen presenting cell and the helper T cell, and they bind to each other. The antigen presenting cell will release signaling molecules called cytokines to turn on this helper T cell. And now you have the helper T cell on, and you get a lot of the helper T cells, and they'll go on, we're talking about humoral immunity now, they'll go on to activate the whole humoral immunity process, which has to do with creating plasma cells and memory cells. And so the specific B cell binds the antigen by the antigen cell receptor. The helper T cell will secrete signaling molecules to actually turn on the B cell. Because remember, helper T cells are the cells that turn on any part of adaptive immunity. So here we're talking about humoral immunity. And then you have expansion and differentiation of B cells, meaning you get a lot of B cells made and it differentiates. So B cells become plasma or effector cells. These are the cells that make antibodies, and they become memory B cells. So differentiation into two different types of cells. Plasma cells or effector cells are the B cells that make antibodies, and memory B cells will also form so that if you are ever exposed to that extracellular pathogen, again, because we're talking about humoral immunity, your body can deal with it. And the outcome of humoral immunity is that B cells secrete antibodies into the blood and lymph fluid, both are fluids, and the antibodies will destroy the extracellular pathogens. That's why antibodies exist. So summary, we have antigen processing and presentation. Then you get helper T cells activated, and helper T cells go on to activate B cells because this is humoral immunity. B cells will secrete antibodies, and it's the plasma cells that are actually making antibodies to eliminate the pathogen. And antibodies will do different things. We'll talk about them. And then you also get memory B cells formed so that they can be ready if you're ever infected again. And this whole process is a big time what happens with people infected with like SARS-CoV-2. So patients infected have antibodies. And so some of the research being done right now is, can we take antibodies from people who've already recovered and inject them into people who are sick so that their antibodies can go on to target the virus? So just keep your eye on that, and I'll talk about it in a second. And now we're going to talk about why are antibodies important. So in humoral immunity, we talked about antibodies, and I just said they destroy cells, but I didn't talk about the details of what they do. So this is a good video, and we'll talk about it.
The main role of antibodies secreted by plasma cells is to tag foreign molecules or microbes for destruction. Binding of antibodies to antigens can trigger several effector mechanisms. Neutralization occurs when antibodies bind to antigens, blocking their activity and making them harmless.
So this is one thing.
For example, antibodies cover the surface proteins of this virus, preventing it from hooking up with the host cell. Other antibodies may bind to toxin molecules on a bacterium. The bound antibodies attract macrophages, which dispose of the invaders by engulfing them, a process known as phagocytosis. Antibody molecules have at least two binding sites, allowing them to form a clump of microbes by binding to two antigens on two different cells. This process, called agglutination, traps microbes and makes it easy for macrophages to capture them and dispose of them by phagocytosis. Precipitation is similar to agglutination, except this process clumps together free antigen molecules dissolved in body fluids. This inactivates them and attracts macrophages, which destroy them by phagocytosis. One of the most important mechanisms of humoral immunity is the activation of complement proteins. Binding of antibodies to a foreign cell triggers complement proteins in body fluids to form a hole in the membrane of the target cell. The hole allows water and solutes to enter the cell. The foreign cell swells, bursts, and dies. Humoral immunity, mission accomplished.
So with humoral immunity, we get activated B cells. Activated B cells will differentiate into plasma cells. Plasma cells make antibodies. And now we're talking about why those antibodies are important in targeting the pathogen. So antibodies can have a lot of different outcomes, as the video spoke about. So I'm going to talk about a few things. Antibodies can do neutralization. So if you look at this image, which I probably should have made bigger, but anyway, if you coat the pathogen or the toxin, they cannot bind cells, they cannot cause infection. So that's one good thing that antibodies do. And then when they neutralize them, you can get a phagocyte, a macrophage to come and phagocytose or eat that pathogen. Antibodies can also enhance phagocytosis, again, which is just called opsonization. They kind of create a tag on like, hey, come do phagocytosis. Complement activation. Remember, we talked about the complement system, and we said in innate immunity, one of the things that the complement system does, complement proteins, is they cause holes in the cell membrane, which causes lysis. This is good if you're trying to kill a cell that's been targeted, a bacterial cell. And agglutination, cross-linking, you can get a bunch of the pathogens by the antibodies to clump together. And when you clump them together, it makes it easier for your immune system, for the phagocytes to go on and eat the bugs before they spread more. Cytotoxicity, antibodies can go on and target the cells for death, and that has to do with natural killer cells. And then finally, you can immobilize and prevent pathogens from adhering. So if the antibody binds the flagella of like a bacteria, then the bacteria can't move. So therefore it gives it more room for different phagocytes to eat it before it causes more infection. Antibodies are found in plasma and serum. They're found in other secretions. We'll talk about them, but to summarize, they're found in your blood. And when we look at blood, if you take a blood sample from someone and you centrifuge it, you spin it really fast, you can separate the different components of blood. I just want to say that the liquid component of blood, called the plasma or serum, is where we find antibodies. So when we say that we're looking at someone's serum to see antibodies, it means that you've spun the white blood cells and the red blood cells down, and now you're looking at the serum. This is where you see the antibodies. When we look at the structure of antibodies, antibodies are globular, and so they're also called immunoglobulins or IG. It's the same thing. If I say immunoglobulin or antibody, it means the same thing, and it comes because when you look at them, they're globular. They're proteins that form a Y shape, so this is why a lot of times scientists draw antibodies like this. And they have constant and variable regions. When you look at the antibody, the bottom of the Y is the variable region, and this is the part that binds different antigens. So this is what an antibody looks like. And there are 5 different classes of antibodies, or 5 classes of immunoglobulins, as we call them: IgM, IgG, IgA, IgD, and IgE. They have the same basic structure, but they're found in different areas in different amounts, and they have different functions and properties. So we're going to go over that. So IgA, IgD, IgE, IgG, and IgM. These are the 5 different classes of immunoglobulins, and IgA is found in secretions. What's in red is what I want everyone to know. IgE has to do with the allergy response. IgG is the most abundant in our serum, and IgM is the initial response of infection. We're going to go over each one right now. IgM is the first antibody that we see produced in response to an infection. So it's the first class produced during a primary response to an antigen or an infection or a pathogen. So when you have a pathogen that infects you, we're going to see a spike in the beginning of infection of IgM. Remember, the antibodies are there to protect you, and its main role is control bloodstream infections. This is where we see IgM. I want everyone to know that IgG, another subclass of immunoglobulins or antibodies, is the most abundant antibodies you see in your blood. And this one is the most abundant circulating class of antibodies during secondary infections or secondary response. So when we look at an infection, you see IgG spike again during the second time we talk about an infection. So I'll mention that. And I do want to mention that IgG is the antibody that can cross the placenta to the fetus's bloodstream. So it's important for protecting the baby, and this is the antibody that's going to the baby. It's not like IgA, for example. And we also see it in colostrum. This is the first breast milk that comes out, usually for a couple of days. And so the baby is getting IgG from the mom just in the beginning of breastfeeding. And it's the most abundant, and we see it circulating during secondary response as well. IgM, the other one, we see a spike of it in an initial infection. The next immunoglobulin is IgA. We see IgA in secretions. Remember, we talked about that ELISA lab and looking at the different IgA amounts in your saliva. You were looking at it in saliva because IgA is the antibody that's in your saliva that's protecting you. It's in mucus, it's in tears, it's in breast milk. This is what I want everyone to know. And when I say mucus, I mean mucous membranes, so gastrointestinal membranes, genitourinary, respiratory tract. So all of these different organs that secrete mucus, in that mucus you see IgA. So IgA is there to protect you. IgE is barely detectable in serum. There's very little of it, but I want everyone to know 2 things about IgA. It's important in protecting you against parasitic worm infections, and it's also responsible for allergic reactions. So when we learn about allergies, we're going to see a lot of IgE. IgD, there's less than 1% of it. I guess the biggest thing is no one really knows what its exact function is. We have some idea that it's part of the B cell receptor, but its exact function isn't yet defined. And when we look at infection, there's a primary response and a secondary response to infection. Primary response is the immune response that happens when you're first exposed to a pathogen or an antigen. So in the primary response, we see IgG and IgM antibodies. Secondary response, when we say that as scientists or doctors, it means that the response that occurs the second or third or 10th time that you're exposed. We just call that the secondary response to a pathogen. It means you've already been exposed to it. And memory cells are formed after you've been exposed to a pathogen. So that's why the secondary response to a pathogen, or the third, fourth, fifth, 10th, we all call them secondary response, is usually much stronger, and you recover much faster if you see the same pathogen again. So if the antigen is encountered again, this secondary immune response is faster, stronger, you see more attacking of cells, longer duration than the primary immune response, meaning your immune system is just there fast and trying to target that pathogen. It's already been prepared. IgM we see in the initial response to an infection in the beginning, and then IgG we see a lot in the secondary response. Your body's making a big amount of it because it's already been prepared. A few terms I want you to know are seronegative and seropositive. Seronegative, sero comes from serum. Remember, this is where we see antibodies. If someone is seronegative, it means that they haven't yet been exposed to that pathogen, so we don't see antibodies in them. And seronegative means when you look at their serum or blood sample, you already see antibodies being made, so they're seropositive for that infection. So if I tell you someone is seronegative for an H. pylori infection, it means that they do not have the antibodies that target H. pylori, which causes stomach ulcers. Titer is the amount of antibodies in serum, and it indicates a previous exposure. So when we say that we see a rising titer in someone, it means that you're seeing antibodies. So a recent infection, if you see a high amount of antibodies and you take a sample every day or every other day, you can tell a lot about someone. A steady but small titer of antibodies means that someone has had an old infection in their body that's still remaining at low levels. And detecting the different amounts and types of antibodies tell your doctor a lot. Are they in the secondary response of an infection? Are they in the primary response? Is it an allergic response? So it's good to have an idea of the antibodies. So that was humoral immunity. Now we're going to talk about cellular immunity. Cellular immunity has to do with T cells and cytokines. And as a summary, cellular immunity has to do with T cells, not B cells. B cells were the ones that produced antibodies. In cellular immunity, we're not forming any antibodies. We're forming T cells that will go on to target cells that have been infected intracellularly. And then this is where we're going to talk about cytotoxic T cells, which are also called CD8 cells, and helper T cells called CD4. And this doesn't look good, so ignore that table for now. The steps in this are, remember, with all adaptive immunity, you need activated helper T cells to go on to activate other cells. So helper T cell will stimulate other T cells to secrete cytokines. Those cytokines will activate cells so that you can get cytotoxic T cell activation. If you get cytotoxic T cell activation, you get killer T cells, which are effector cytotoxic T cells, which cause cells to die. Because if you've been infected with a virus, you'd rather that cell die than that cell staying as a breeding ground for that virus. You get memory cytotoxic T cells, and you get activated macrophages to go on and target the pathogen even more. And if the same pathogen infects again, memory T cells are there to quickly respond to that intracellular pathogen, such as a virus. The cytotoxic T cell, again, as a reminder, it defends against pathogens that have entered body cells or intracellular pathogens. They're the only T cells that actually kill infected cells. So when we say cytotoxic T cells, these are cells that kill infected cells. They're not helper T cells. So here you have a cytotoxic T cell. It binds to an infected cell and it becomes activated. Remember, it becomes activated because you get cytokines released that activate it. Then the activated cytotoxic T cell will cause the cell that's infected to die. So it'll become a killer T cell. It will kill this cell. Usually it does it by causing formation of different proteins that cause holes in the infected cell. Okay, actually, here's a good video.
Now let's look in more detail at cell-mediated immunity. Cytotoxic T cells are the main warriors of cell-mediated immunity, which attacks body cells that have been altered by infection or cancer, or transplanted cells that are foreign to the body. An infected, cancerous, or transplanted cell displays foreign antigens on its surface. T cell receptors on specific cytotoxic T cells recognize and bind to specific antigens on cells. Cytotoxic T cells respond to this contact and to interleukin-2 sent out by helper T cells by producing the protein perforin. Perforin molecules are released from the cytotoxic T cell and attack the membrane of the infected cell, punching holes in it, which allows water and solutes to enter. The cell bursts and is killed. In this way, cytotoxic T cells deprive pathogens of.
So that's what I was talking about, cytotoxic T cells. When activated, they go on to kill cells. So they become effector killer T cells. So the process, again, with cellular immunity, like humoral immunity, first of all, you have to have some sort of infection. So the infection by the pathogen or the pathogen's antigen. So the antigen is processed, it's presented by antigen presenting cells that are floating around. Antigen presenter cells will go on and activate helper T cells, and then helper T cells will go on to activate cytotoxic T cells, for example. So the antigen presenting cell, with presenting the antigen, will bind specific cytotoxic T cells by its T cell receptor. It will secrete cytokines, which will activate the cytotoxic T cell, and the cytotoxic T cell now is ready to become a killer T cell and kill the, for example, virally infected cell. And you also get memory cytotoxic T cells. So if you're ever exposed to that virus, again, if we're using viruses as an example of an intracellular pathogen, you have memory cytotoxic T cells. So you destroy abnormal cells. It's the same idea with cancer cells. So if you have infected, virally infected or tumor cells present, the antigen is presented. Remember, the antigen is presented on MHC molecules. The killer T cells will recognize this complex via their T cell receptors, and they'll go on to release different molecules such as perforins or proteins that will cause that cell to die or cell death. Another thing we can get from cell-mediated immunity, so that was activation of cytotoxic T cells, is cell-mediated immunity can activate macrophages. Macrophages are a great cell because they do phagocytosis. So they phagocytose or eat pathogens. So again, with activated macrophages, first you have antigen processing and presentation. Helper T cells are activated, and those helper T cells now, instead of activating cytotoxic T cells, they can go on to activate macrophages. And the outcome is, if you have activated macrophages, you get improved phagocytosis. Here, you're not getting any memory cells, unlike cytotoxic T cells. You're just getting activated macrophages, which is a great thing. And in summary, with cellular immunity, with all of them, you have antigen processing and presentation as the first step. Then you have helper T cells activated. And then you get cytotoxic T cell activation and macrophage activation. And the outcome are cytotoxic T cells will become killer T cells when activated, and they'll cause cells that are infected to die, which is what we call apoptosis. This process will also lead to activated macrophages. Activated macrophages means you get improved phagocytosis. So this is cell-mediated immunity. And so we talked about in both adaptive in both humoral immunity and cellular immunity of adaptive immunity. This is a very long lecture. You have antigen processing and presentation. then they activate helper T cells, and then helper T cells will either go on to activate humoral immunity or cellular immunity. Humoral immunity has to do with B cells. B cells attack extracellular pathogens. T cells attack intracellular pathogens, including cancer abnormal cells. Now, I just want to end the immune system by talking about HIV. HIV, as many of you know, is a very devastating virus, and the reason is that it causes someone to lose their immune system. To take it one step further, the reason why people with HIV infections don't have an immune system, I mean, over time, you basically lose your immune system, is because the HIV virus infects helper T cells. Remember how when we talked about adaptive immunity, we said all adaptive immunity, whether that's humoral immunity or cellular immunity, is activated by helper T cells. Well, if you have a virus that's infecting helper T cells and causing them to die, you will no longer have humoral immunity and you'll no longer have cellular immunity. So people with HIV infections basically lose their adaptive immunity over time, and they develop acquired immune deficiency syndrome, which is AIDS, and so they lack a functioning immune system. And I want everyone to know that helper T cells activate all of adaptive immunity. They activate B cells and they activate T cells, so in humoral immunity and cellular immunity. And finally, we've been talking about human or animal immune systems. Bacteria also have an immune system of their own, and that is CRISPR. So we learned all about CRISPR already in biotechnology, or one of the lectures, I forgot. CRISPR is a system that we see in around 50% of bacteria. I'm sure it's in more, but it's just not been discovered because it's still a developing area of research. The CRISPR-Cas system is a system that bacteria have in them so that if bacteria are infected by viruses, remember bacteria can get infected by viruses, which we call bacteriophages. The bacteria that have CRISPR can go and cut the virus's genome material out of their own genome, because that's what viruses do. And by doing this, they get rid of the viral infection. So this is their adaptive immunity, but they also form a memory if they're infected by that virus again. So the CRISPR-Cas system is a form of adaptive immunity specifically. So it protects bacteria from viruses and other mobile or movable genetic elements. And so remember that with the CRISPR, you have one protein that cuts out the viral DNA, one that guides it to it. But the reason why this is a form of adaptive immunity in bacteria is because bacteria will remember the virus with another exposure, so it's a form of adaptive immunity. And finally, to summarize this really very long lecture that if we were doing in class, we would probably do in like a week and a half. When we look at adaptive immunity, there's cellular immunity and there's humoral immunity. Humoral immunity has to do with B cells, which go on to protect you against extracellular pathogens, bacteria, toxins, foreign red blood cells. Cellular immunity has to do with intracellular pathogens such as virally infected cells, intracellular bacterial infections, cancer cells. And so that's cell-mediated immunity, which has to do with T cells. Humoral immunity has to do with B cells. And all of this cannot be activated until helper T cells are activated. Helper T cells are activated by antigen-presenting cells. And then once helper T cells are activated, they go on to activate B cells or T cells, depending on what route of infection they're going in. And all of this activation has to do with cytokines, which are signaling molecules or specific interleukins that are released. So this was the whole immune system that we did. I hope you guys can put everything together now. So in the other lesson, we did innate immunity and we did adaptive immunity today. And I want everyone to keep in mind that all of this is happening at the same time. It's not bits and pieces. Your innate immunity is protecting you, adaptive immunity, humoral immunity, cellular immunity, all of these things are happening in you. So I hope you guys take time to review this and put everything together, and good luck on all of this information.